The story of our planet isn't just written in mountains or seas, it's hidden in particles smaller than a grain of sand. When you look into a microfossil, you are not just seeing a shell, you are glimpsing a memory, a fragment of an ancient ocean. And for over 4 decades, one man has dedicated his life to decoding those tiny whispers from Earth's past. Professor Pradeep Kumar Kathal, renowned micropaleontologist, researcher, and former head of the Department of Geology at Dr. Hari Singh Gaur Vishwavidyalaya. Through years of study, teaching, and discovery, he's brought to life the mysterious world of microfossils, revealing how these minute forms hold the secrets of shifting climates and vanished seas. In this dialogue, we dive into his journey, one that shows how even the smallest traces of life can tell the greatest stories of our planet. Tiny fossils, big stories. From ancient oceans to modern climate change. The smallest clues unlock the largest mysteries. Today, we sit down with Professor P.K. Kathal to explore the unseen world that reshapes Earth's past and also plan its future. Professor Kathal, thanks a lot for joining us.
Thank you.
For viewers new to the field, could you please introduce yourself and your journey into micropaleontology?
Well, I have been a student of geology, paleontology, and micropaleontology, and have just retired 2 months back as senior professor from the Department of Applied Geology of Dr. Hari Singh Gaur University, Sagar.
So, sir, if you had to explain micropaleontology at a family dinner, no technical jargon, but with people who are completely new to this word, how would you describe it?
In fact, microorganisms may be preserved in sediments which are converted into rocks later on. We separate them from the rock and we try studying their morphology, their conditions in which they lived, or we call it ambience, and how other information can be retrieved which are useful in geology, and therefore micropaleontology has gained importance in the last 40-50 years.
Well, this field certainly sounds very interesting, but still, some of my friends ask that why should someone outside geology care about microfossils at all?
Well, I think students pursuing their bachelor's and master's in geology and botany, they always wonder how the life has started and has evolved, and how do we calculate the age of rock and different stages of evolution. So I think it is interesting for those who are pursuing career in geology and botany.
Well, this certainly cleared the doubt. I would like to ask that what was the wow moment that first hooked you up into this subject when you were in the bachelor's, like we people?
In fact, since my childhood, I was very much interested in the study of animals and plants. And when I started my graduation in geology, I studied paleontology, and there I came across how simple life of animals and plants from bacteria has evolved to the complicated and advanced life by interaction with the environment. And that fascinated, and I immediately thought that I will make my career in this particular branch.
So, sir, what exactly are microfossils? Where do they come from?
Actually, once living protists, microscopic animals, other animals, plants, which are less than 4 mm in size, and they get fossilized, we study them under microscope. And therefore, you can say it is a technique-based branch of study of invertebrate organisms which are preserved as fossils.
Okay, so all those living microscopic organisms.
Once living microscopic
Okay.
organisms which have been transformed as fossils due to their preservation in the rocks.
Okay. So, do we need a special form of microscope or we can observe it?
Yes, this is a very interesting question. In fact, we require zoom stereo microscope where we can have depth perception so that we can view the organism as a whole. I mean, the periphery and the apex, everything should remain in focus. And there is an arrangement where we can zoom the image and we can also take pictures.
Okay, so these things have become really interesting now. So we know that it's not just any microscope or any fossils. We study about microfossils and we use special techniques and special microscopes for that. So could you tell us how does something so tiny as a microfossil help us determine something so large like the geologic time?
In fact, these tiny organisms, they have shells which are preserved in rocks. And we have a principle that says the older rocks at the bottom and younger rocks at the top, if the sequence is not disturbed. So we collect these tiny organisms and we find out relative ages. Then we go for accurate ages and that gives us the dating of rock. And on that basis, we develop classification of rocks in geological time that is called biostratigraphy. We can also correlate rocks of one area with the other area on the basis of the biota which is preserved in the rock. Similarly, we can work out the environment in which the rock was deposited. Since these are microorganisms, they are really very sensitive to even minute micro changes in the environment, and that is reflected in their morphology. So we have a kind of a process in which we understand how they can best indicate the environment, and moreover they have been widely used in exploration projects like oil exploration and coal exploration, and they have minimized the cost of exploration as well as the time of exploration, and that helps us in developing on a faster pace.
So, sir, there is a friend of mine who is studying geology. He recently went on a trip and he was mesmerized about rocks and sediments. He was obsessed about them. I also know a fact that there is a principle on life that Earth changes in recognizable steps through time. So my friend asked me that how can we study these rocks, these sediments to timestamp these various recognizable steps?
As I said earlier, the life has evolved from very simple unicellular form of life to the multicellular life which we find, including man. And it has not in a continued process. It has life has evolved in different breaks in time, and that period we call extinction period. And we can say that overall period for evolution of life is about 540 million years from where the microorganisms appeared first. So regarding the early stage of evolution, only microorganisms and microfossils can guide us. Therefore, their importance in earliest form of evolution of life is there.
But sir, I still find these concepts actually a little hard to grasp. So can you give me a simple analogy like a library catalog or barcode or something so that how does these microfossils help place layers in order?
As I said that layers of rocks naturally have deposited one over another during a particular period of time. And if we consider the evolution of organisms, naturally we will find the earliest animals in the lowermost layers, followed by intermediate type of life and the youngest life. So this is how we can barcode. If you show me a fossil, I can work it out in which particular period it lived on the earth or whether it is continued still or not. And on that basis, we can go for relative ages first. We cannot go straight to absolute ages or accurate ages. Relative ages. It means that this fossil must not be older than this and this other fossil must not be older than that. Then we correlate it with the time period on the basis of geological time scale, and we can actually date the rock. And not only this, we can correlate rocks of one area with the other, as I said earlier also, on the basis of these tiny organisms. So they not only help us dating, but also help us in correlating rocks of one area with another. Similarly, if we work out their sequences across the continent where the climate zones were different, we find their equal ancestors are were deposited in the rocks, which through their course of evolution we can work out.
So we got to know that these microfossil samples are so important, and we can correlate them across the globe. So now the question arises that how is it collected? How is it studied? So, sir, could you please walk us through a samples journey from the collection, from the field collection to the microscope in which we are going to study it? What happens in each step?
We visit the rocks in the field and we collect samples from each layer from bottom to top. We number the samples, location, and we bring them in the lab. Then we break it and we try pulverizing the sediment by using different kinds of soluble substances starting from water or mild detergent powders, kerosene, and also some light acids depending upon the composition of rock and the composition of shell of the particular microfossil. After pulverizing, it means after converting it back into the sediment state, we sieve them through a set of sieves which have sieves size of different diameters, and we collect fractions from each sieve and then we watch them under the microscope. If the rocks are from Phanerozoic time, it means younger than 400 or nearly 500 million years, then we will find that one or the other kind of microfossil is there depending upon the environment. It could be freshwater microfossil or marine water microfossil.
So this was about collection of those specimens. What about when it reaches the microscope? What features tell you this species, this age, this environment when you look through the microscope?
Well, we can differentiate different groups of microfossils on the basis of their composition of shells. And we can differentiate arthropods from foraminifera, we can differentiate diatoms, we can differentiate calcareous nanoplanktons. And this is how we first find which group is prevailing. Then once the number of groups have been decided, then we develop microfaunal slides in which we keep differently looking species in different sectors, and then we go for its taxonomy. And once the taxonomy is determined, then we work out the age and the environment in which that fossil or that organism was first living and which has been converted into a fossil. So this is the simplest process that we follow.
You just mentioned diatoms. So are there also any other groups that you study? And what clues does each group give?
Well, as I said, the microfossils, we can get different groups. We broadly divide them into two. One is called organic walled microfossils and second is called mineral walled microfossils. Organic walled microfossils are generally the fossils of pollen and spores, which are also called as dinoflagellates. And in case of mineralized wall, we have foraminifera, which are calcareous or arenaceous. We have diatoms, which are siliceous. We have radiolaria, which are also siliceous. Ostracods, which are chitinocalcareous. So all of them have different composition, and by judging their composition either under a high magnification microscope or under scanning electron microscope, we can work out their composition and also the group, and then we go for the detailed taxonomy of that particular group.
So this was about the sample collection and also how we study the sample. Sir, I would now like to ask you that how does computers and other instruments help you
identify the specimens or spot patterns?
Well, the microfossils are important because of another reason, that they occur in great abundance. And even smallest fraction of sample can give us thousands of microfossils, or maybe millions of microfossils, or billions of microfossils. And when we have that much of data, naturally we need some mathematical treatment to these number of species, and we go for different statistical analysis, and we can also make use of computers. And my students are developing certain methods here. Elsewhere also students are developing using artificial intelligence, in which the primary data will be fed, and then identification work will take little time for further analysis.
Sir, could you tell us how does microfossil help study and reconstruct the past climates?
As I said in the beginning, they are very tiny, and most of them are unicellular. Therefore, they are very sensitive to even minute environmental changes. And change in environment is reflected in their morphology, and also they manifest in certain other ways. We have developed certain methods by which we can uh, pick the change in the environment, and therefore we can work out how the environment was gradually changing in geological period of time.
And are they also helpful in studying monsoon of the past?
Yes, it is very interesting. And last 30 years, micropaleontologists have developed a study of paleomonsoon precipitation, which is in terms of 1 kilo a year, 5 kilo a year, 10 kilo a year, and 20 kilo a year. In fact, in a country like India, where we have mammoth population, our economy is based more on the agricultural produce. And spell of bad monsoon means lot of problems in the. So we need to learn how the monsoon is going to behave in future. For that, we cannot rely on meteorological data because it is only of last 200 years, while geological phenomena, they take thousands of years. So we rely on these tiny organisms, not only foraminifera, but all other microfossils, which act as proxy in understanding the change in climate. And on that basis, the scientists have used the data in order to understand whether the monsoon was cyclic in nature, or whether it is a kind of erratic in nature. And I am happy to tell you that cyclicity has been noted down, and we are very sure that in future we will be predicting monsoon in terms of 50 years or 100 years.
So we just saw that by studying the past climate and past monsoons, we can extrapolate that data and we can even predict the climate of the coming 50 years. So sir, now I would like to ask you that where do these microfossils help guide in decision making in terms of energy and minerals exploration?
Well, you see that every country is paying a lot on the oil and gas. And these tiny organisms in the form of fossils, they guide us whether the rocks were, or the conditions were congenial for conversion of organic matter into oil or not. If we look at the geological record, we find that the rocks of tertiary period, starting from 66 million years to around 2.6 million years ago from now, they are holding world's 90% of oil. In India also, we have such. So without use of microfossil, we take a lot of time in exploration by drilling and then checking out. And but if we use microfossil, we can ascertain the age of rock, whether it is tertiary or not. Second, we can also see whether the rocks were deposited in the correct type of environment where the organic matter could be converted into oil. And there are certain species of microfossils which indicate these environments. So not all the tertiary rocks are holding oil. Only some part of tertiary rocks we have to pinpoint. Tertiary rocks, they are having thickness of thousands of kilometers. We cannot waste time. and money. So in a way, by using microfossils, we are making the project time and economically we are making the project viable in terms of time and money. So this is how we say that if we use micropaleontology, we can save time as well as money, both on any project.
Okay, it's very interesting to know that how these little things, microscopic, and then they are helping us in so vast extents. We just discussed that they can use to predict climates of 50 years. And now we also got to know that we can enhance the quality and also the economic feasibility of many vast sector projects. So I guess this also adds to the importance of studying microfossils.
So, sir, can they also help monitor pollution changes in water bodies like lakes and rivers?
Well, as I said earlier, twice, that they are very small and sensitive. So, under normal environmental conditions, we find their normal population as well as diversity. While if the environment is polluted, naturally either the population will go down or there will be only few species which can withstand change in the environment. And therefore, we find that the present day environment can be monitored, whether it is freshwater or marine water. Now, the second point which I am going to emphasize is that we are depending the population is depending in most of the countries on seafood and also on the other kind of food which is kind of fishes and all that. And if we do not pay attention and our environment goes against the habitat of different species, it will make impact on the production of seafood, which will be very bad for the economics of the whole world. So all the countries are monitoring and observing the coastal environment so that they can get more and they can keep their environment healthier, so that the future coming generation also get the same kind of sea produce.
And sir, could you also give a real example when a microfossil based insight changed a project or a budget or a timeline?
Well, I tell you a very interesting story. Geologists were looking for oil in Cambay Basin in Gujarat, and they ascertained the age of rocks and the environment of the deposition on the basis of foraminifera. Then they found that there are so many shells of ostracoda, and most of the shells were closed because ostracods are tiny bivalve animals, and if the rate of sedimentation is too high, then they don't get time after their death, they do not get time for opening of the shells. So they are buried as such. The percentage of these closed carapaces in Cambay Basin was of the order of around 90%. And that has given us the idea that it could be a very large pool of natural oil, and it is one of the largest pool of oil in India. And this is how foraminifera and ostracoda has helped the scientists to understand within a short period of time they could complete the project.
So we just saw an example, a real life example where a microfossil based insight was so crucial for a project. Sir, can you tell me what makes India a rich laboratory for studying micropaleontology? Is it Himalayan basins? Is it Deccan related sediments? Or is it the coastal cores?
You see, the development of geoscience in India is of very high order. And also in micropaleontology, the environment of research is very conducive. There are so many organizations. Some are working on Himalayan regions, some are working in the different coastal areas and also in deep sea. There are organizations like National Institute of Oceanography where you can get repository of sediment cores of different Himalayan areas. Coastal area is generally covered by the National Institute of Oceanography. Birbal Saini Institute of Paleosciences in Lucknow, they are also working out various territorial or terrestrial kind of deposits. They are maintaining the log. So we have complete log right from Himalayas down to the ocean, and the repositories are maintained by different organizations like Oil and Natural Gas Corporation, Coal India Limited, National Mineral Development Corporation, and from there we can get core samples and we can use the samples.
So, sir, since you specialized in foraminifera, could you share us a story in which a foraminifera-based insight actually impacted a project?
Well, it reminds me a very interesting story. In fact, I was working on some foraminifera, which are called as low oxygen foraminifera. And then I found that they are also facultative anaerobes, that they can develop certain changes in their morphology to withstand change in the uh, concentration of oxygen towards lower side. When the oxygen was low, the number of pores and the diameter increased. And in the same species, in the same specimen, we find that periodically when the oxygen level was going up, the same number and size was reducing. So it means that it has developed a very, I mean, technical way to withstand change in the concentration of oxygen. This I used in case of Miocene foraminifera, and I found that it is true. And in Indian petroliferous basins, scientists are using this criteria in demarcating those sedimentary zones where such low oxygen species are there, and they are also called as morphological tool to indicate low oxygen.
So, as India is a largely an agriculture-based country, so could you tell us how does studying about monsoon and climate from these microfossils help us understand the security risks related to agriculture in India?
Well, this is very important. In fact, poor monsoon means lowering of our economy. And as I said, India's population cannot cope up with poor monsoons. If we are able to predict monsoon in terms of 25 years bracket or 50 years bracket, we can prepare ourselves when our monsoon is going to be poor, and we can make adequate arrangement for other irrigation technologies so that we do not have to pay heavily because of poor monsoon.
So, sir, could you share us an experience where you collaborated with an oceanographer or an archaeologist?
Well, I have worked with a few organizations, and I have also worked abroad, particularly in Japan and Germany, where we have done some collaborative research. In India, I had a project with the Geological Survey of India, and I have collaborative research papers from scientists from Indian Institute of Oceanography, from Birbal Saini Institute of Paleosciences, and other universities.
So, sir, could you tell us how do microfossil timeline pair with tools like radiocarbon dates or magnetic signatures to build a stronger story?
Well, we have two sets of environmental conditions. One is called shallow water environment in the ocean. Second is called intermediate to deep water environment. As I said earlier, the calcareous microorganisms, they have a limit up to, say, calcium carbonate compensation depth, below which we get only siliceous type of microfossils. We have observed that radiolarians, they have got a very strong connectivity with the change in the magnetism, and the paleomagnetic reversal has been correlated with the change in the species in radiolaria, and still we are not able to comprehend the real reason, but it is working very fine in order to classify rocks and also the environment of different sets of conditions.
So, sir, what are the biggest challenges in this study, and also how do you cross-check your interpretations?
You see, we have two types of assemblages. One is called biokinos, where the organisms get fossilized wherever they are living, while after their death, if they get fossilized elsewhere, they fall under the category that is called thanatokinos. So we are always uh, looking for those assemblages which are bioconos so that they give us the true picture of sedimentation, and if they have been carried over or transported by the current with the sediment to some other place, then we cannot ascertain the real climatic condition which prevailed during the time when they were living in the environment.
I think the viewers and sir will agree that science mostly becomes a detective's work. So, sir, could you tell us an experience in which you found the most surprising clue in your detective work?
Yes, well, I remember one occasion where I found that one species of foraminifera named as Astrodotelia trispinosa. It has three cylindrical spines which are equiradii in their length, and it was occurring on mud flats where no other foraminifera could. We could think that any foraminifera can live because foraminifera got buried under the mud. But by virtue of its spines and the diameter of its spines, it was able to survive and it dominated. So therefore, we designated this species as a marker of mud flats, and it has been very widely used by other scientists in working out the environment of the coastal sediments in India.
Have you had any moments where the data challenged your expectations? And during those moments, how do you handle this situation?
Yes, I compared the foraminiferal studies, foraminiferal species from nearly 26 stations from west coast of India and east coast of India. The stations were stretching over 5,500 kilometers, and I found that the data was so enormous, and I wanted to compare the conditions in the Bay of Bengal and the Arabian Sea, which is also called as geographical province. I found that only 26 species were common on the basis of a statistical treatment that took me around 10 years to complete because the data was enormous. And after I compiled it, then our university computer center, they developed a special program for this statistical treatment, and on the basis of which we could find out that the oceanographic conditions are very different, and the fauna on the west coast of India is falling in the East African affinity, and on the other hand, the fauna of the Bay of Bengal is falling in the Indo-Pacific affinity. So that makes the marine food life is also very different in the two conditions, and therefore we find more fishes on the west coast and less fishes on the east coast.
So, what are the responsibilities that come up when we study Earth's past, and how do they influence today's and future's policy decision making?
In fact, scientists are working on various projects that are related to the oceanography, monitoring of the coastal environment, and also the civil engineering constructions which are taking place on the coastal areas, and also underwater projects, for example laying pipelines, laying cables and other kind of structure. And also other countries are sometimes dumping the nuclear waste in unauthorized manner in no man's land. But suppose we do not study oceanographic characters and the water is coming from that area to our area, then we will have our own environment is spoiled. So they're continuously monitoring on the basis of international cooperation is going on, and still every country has to pay attention to oceanographic researches, and India is very advanced in this field.
So, sir, after studying micropaleontology for all these years, what breakthroughs do you see on the horizon for this field? Is it related to AI, or is it related to high image resolution, or is it related to new drilling methods?
Well, very recently I have attended one national conference, and I found that the scientists are working in different fields where they are utilizing information based on isotope studies. They are utilizing information based on genome studies in identification. And even I found that some papers were presented where use of AI was promoted in order to reduce the time in identification of different species. And I'm very sure in next 10 years, all these studies will be culminating to, and India, I'm telling you, I was impressed that India is, just at par with other international standards so far as these researches in micropaleontology and such kind of studies are concerned. As regard drilling, I cannot tell you much because it's a very different kind of mechanical process where my use of micropaleontology is really not appreciated.
So let's say we revisit this conversation in next 10 years. What do you think will be changed in how we see micropaleontology in Earth's past climate?
As I said earlier, also micropaleontology is technique-based branch, and you see always there are new breakthroughs in technologies in every field. So I believe that different technological breakthroughs which are coming, and I foresee them in micropaleontology, they will be devised to save time and money, and we will be more time and money viable in terms of such kind of micropaleontological investigation. And I also foresee the use of AI where the scientist has to simply put a species under the microscope and the computer will give you not only the age of the that fossil but also the associated environmental conditions and other peripheral information. So I think that next 10 years the artificial intelligence is really going to take place and it will save a lot of our time.
So what are the big questions that keep you excited or let's say awake right now regarding the field of micropaleontology?
You see, growth of knowledge is always a spiral. And we find that more we study, more questions arise. So I'm really afraid that we have still not explored the sea sediments, which are covering very large area of the earth, and how we are going to incorporate all the information. That keeps me little worried about it.
So this is perhaps the most repeated questions that was asked to me when I was researching about the questions to various people, especially students. So sir, could you tell us for the students watching, what skills matter the most when we for someone who is beginning to study micropaleontology? Is it careful observation? Is it statistics? Is it GIS, lab methods, or writing?
Or
All of them.
everything is important. If you make mistake right at the time of collection of sample, or if you make mistake in computation or statistical analysis, the results are never going to be accurate. So you have to pay attention to all stages.
Another most repeatedly asked question. So, can you suggest some low-cost tools or resources or some open resources for people who are beginning to study micropaleontology so that they start seeing things like a micropaleontologist?
In fact, we require basic facilities like the sieving of sediments, pulverization equipment, maceration techniques, a good stereo zoom microscope or SEM, and also some analytical facilities. We do not require in the beginning very large or very highly sophisticated instrumentation. But if we are going for isotope analysis or some kind of genome analysis, we definitely have to seek help of already established institutions because they are very costly affairs. And in the beginning, we cannot procure them.
Okay, now's the time for the lightning round. It's going to be consisting of questions that are fairly one word and simple and straightforward to answer. So, sir, one microfossil group you never get bored of and why?
Foraminifera because it has nearly 36,000 living and dead species, and half of them are found as fossils.
Okay.
Field or lab? Where do you feel most at home?
I think we have to pay more attention when we collect samples because that make mistake that we I mean, that we make in the field cannot be corrected.
Okay, a misconception about fossils you wish would disappear.
I think uh, the answer lies in more and more ecological studies of foraminifera, particularly in vivo, that will throw light on their utility when they occur as fossils elsewhere.
If viewers remember just one idea from today's conversation, what should it be?
Well, I would like to conclude it in this way, that study of small organisms is not small.
So this concludes today's dialogue with Professor P.K. Kathal. Thank you, sir, for joining us in our conversation and seeing the world of micropaleontology with a new lens.
Okay, thank you. I enjoyed talk with you.